Hello students. Welcome to Rush Learning Hub. In this chapter, we are going to explore one of the most fascinating topics in biology, the cell, which is often called the building block of life. Have you ever wondered what your body, a tree, a bird, a butterfly, or even a tiny microorganism has in common? Although living organisms differ greatly in size, shape, and complexity, they all share one important characteristic. They are all made up of tiny units called cells. Cells are so small that most of them cannot be seen with our naked eyes. Yet, these microscopic structures are responsible for every activity that takes place inside a living organism. Whether it is breathing, digestion, growth, movement, reproduction, or responding to the environment, everything ultimately depends upon the proper functioning of cells. In fact, every living organism begins its life from a single cell. Through repeated cell division, this single cell gives rise to millions and eventually trillions of cells that form a complete organism. This shows how important cells are for the existence and continuity of life. That is why cells are known as the fundamental or basic units of life. In this chapter, we will learn how cells were discovered and how microscopes helped scientists observe the invisible world. We will study the structure of cells and understand the differences between plant cells and animal cells. We will also explore various cell organelles and understand how each organelle performs a specific function, just like different departments of a factory. Further, we will learn about the nucleus, DNA, chromosomes, and genes, which are responsible for heredity and control of cellular activities. We will also understand how substances move across the cell membrane through diffusion and osmosis. Another important topic in this chapter is cell division, through which organisms grow, repair damaged tissues, and reproduce. Finally, we will revise important concepts, scientists, and exam-oriented points that are frequently asked in school examinations and competitive exams. By the end of this chapter, you will understand how cells work together to form tissues, organs, organ systems, and ultimately a complete organism. In simple words, understanding cells means understanding life itself. So, let us begin our exciting journey into the microscopic world and discover why the cell is rightly called the building block of life. Let's get started. Hello students. Before we study the different parts of a cell, let us first understand a very important question: what exactly is a cell? Look around you. We see plants, animals, birds, insects, and human beings. They all look different from one another. Some are very small, while some are extremely large. Yet, all living organisms share one common feature. They are all made up of tiny units called cells. A cell is known as the basic structural and functional unit of life. But what does this statement actually mean? The word structural unit means a building block. Just as a building is made up of bricks, every living organism is made up of cells. Similarly, the word functional unit means that all life activities are performed by cells. Activities such as nutrition, respiration, growth, excretion, reproduction, and response to the environment ultimately occur at the cellular level. Therefore, a cell is not only the building block of the body but also the unit that performs all life processes. A cell is the smallest unit of life capable of carrying out all essential life activities independently. Now let us look at the two major categories of organisms based on the number of cells they possess. The first category is called unicellular organisms. The word uni means one. Therefore, unicellular organisms are made up of only a single cell. Examples include bacteria and amoeba. Although these organisms consist of just one cell, that single cell performs all life activities. It obtains food, respires, grows, responds to the environment, and reproduces. This shows how remarkable a single cell can be. The second category is called multicellular organisms. The word multi means many. Multicellular organisms are made up of a large number of cells. Examples include human beings, trees, dogs, birds, and most plants and animals. Unlike unicellular organisms, the cells in multicellular organisms perform specialized functions. For example, nerve cells conduct impulses, muscle cells help in movement, and blood cells transport oxygen. This division of labor makes multicellular organisms more efficient and complex. Now let us understand how cells are organized in multicellular organisms. A single cell alone cannot form a complete human body. Cells first group together to form tissues. A tissue is a group of similar cells performing a specific function. For example, muscle tissue helps in movement. Different tissues combine to form an organ. Examples of organs include the heart, lungs, stomach, and kidneys. Several organs work together to form an organ system. For instance, the digestive system includes the mouth, stomach, intestine, liver, and pancreas. Finally, all organ systems work together to form a complete organism. Therefore, the organization of life follows a simple sequence: cell, tissue, organ system, organism. This hierarchy helps living organisms perform complex activities efficiently. Students, remember this important exam point. A cell is the basic structural and functional unit of life. This is one of the most frequently asked definitions in examinations. Also remember, unicellular organisms contain only one cell. Multicellular organisms contain many cells. Cell, tissue, organ system, organism. With this understanding of cells, we are now ready to explore how scientists first discovered cells and how microscopes opened the door to the fascinating microscopic world that cannot be seen with our naked eyes. In the previous slide, we learned that the cell is the basic structural and functional unit of life. But have you ever wondered how scientists discovered cells when they are too small to be seen with our naked eyes? To answer this question, we need to understand the invention of one of the most important scientific instruments ever created, the microscope. Before discussing the discovery of cells, let us briefly look at the origin of life. Scientists believe that life began on Earth nearly 3.5 billion years ago. According to scientific theories, the early Earth had conditions very different from those we see today. The atmosphere contained gases such as methane, ammonia, hydrogen, and water vapor. Over millions of years, simple organic molecules formed in water and gradually gave rise to the first living cells. These primitive cells were extremely simple compared to modern organisms. Through evolution and natural selection, these early life forms gradually developed into the tremendous diversity of living organisms that exist today. However, because cells are microscopic, scientists could not study them until the invention of the microscope. Now let us move to one of the most important events in the history of biology. In the year 1665, an English scientist named Robert Hooke made a remarkable discovery. Hooke took a very thin slice of cork and observed it under a microscope that he had improved himself. Cork is obtained from the bark of trees and is commonly used for making bottle stoppers. When Hooke looked at the cork under his microscope, he observed many tiny box-like compartments arranged closely together. These compartments reminded him of the small rooms used by monks in monasteries. The Latin word for such small rooms is cellula. Therefore, Hooke named these tiny compartments cells. This was the first time the term cell was used in biology. It is important to remember that Hooke actually observed dead cells because cork is made of dead plant tissue. He could only see the cell walls and not the living contents of the cells. Even though his observation was limited, it marked the beginning of cell biology. Students, this is a very important exam point. Robert Hooke discovered cell S in 1665 by observing a thin slice of cork under a microscope. Now the question arises, what exactly is a microscope? A microscope is an instrument used to observe objects that are too small to be seen with the naked eye. Microscopes work by magnifying tiny objects, making them appear much larger than their actual size. The earliest microscopes were simple, but modern microscopes are highly advanced and allow scientists to study structures at the cellular and even molecular level. There are two major types of microscopes that you should know. The first is the light microscope. A light microscope uses visible light and a system of lenses to magnify objects. It is commonly used in schools, colleges, hospitals, and research laboratories. With a light microscope, scientists can observe cells, tissues, and many microorganisms. However, there is a limit to how much a light microscope can magnify because visible light has a limited wavelength. To overcome this limitation, scientists developed a more powerful instrument known as the electron microscope. Instead of light, an electron microscope uses a beam of electrons. Because electrons have much shorter wavelengths than visible light, electron microscopes can produce images with extremely high magnification and resolution. They can magnify objects hundreds of thousands or even millions of times, allowing scientists to observe detailed structures inside cells. Thanks to electron microscopes, scientists have discovered many cell organelles that were impossible to observe with ordinary microscopes. Now let us understand two important scientific terms often associated with microscopes: magnification and resolution. Magnification refers to how much larger an object appears compared to its actual size. For example, if a microscope magnifies an object 100 times, the image appears 100 times larger than the original object. Resolution, on the other hand, refers to the ability to distinguish two closely placed points as separate and distinct. A microscope may have high magnification, but if its resolution is poor, the image will appear blurry. Therefore, both magnification and resolution are important for obtaining clear and useful images. Students, remember these key exam points. Robert Hooke discovered cell S in 1665 while observing a thin slice of cork. The word cell comes from the Latin word cellula, meaning small room. A microscope is used to observe objects too small to be seen by the naked eye. Light microscopes use visible light, whereas electron microscopes use electrons. Magnification increases the apparent size of an object, while resolution determines the clarity and detail of the image. The invention of microscopes opened an entirely new world that was invisible to the human eye. It allowed scientists to study cells, microorganisms, and the fundamental structures of life. Now let us move inside the cell and study one of its most important structures, the cell membrane. Every living cell is surrounded by a thin, delicate, and flexible covering known as the cell membrane, also called the plasma membrane. The cell membrane forms the boundary of the cell and separates the internal contents of the cell from the external environment. Although it is extremely thin, it performs several important functions. It protects the cell, gives it shape, and regulates the movement of substances into and out of the cell. One of the most important characteristics of the cell membrane is that it is selectively permeable. What does selectively permeable mean? It means that the cell membrane allows certain substances to pass through it while preventing others from entering or leaving the cell. For example, oxygen and nutrients can enter the cell when required, while waste materials can move out. At the same time, harmful substances are prevented from entering the cell. Because of this property, the cell membrane helps maintain a stable internal environment inside the cell. Now let us understand how substances move across the cell membrane. The first process is called diffusion. Diffusion is the movement of particles from a region of higher concentration to a region of lower concentration until they become evenly distributed. Let us take a simple example. Imagine that a bottle of perfume is opened in one corner of a room. Initially, the perfume molecules are concentrated near the bottle. After some time, the fragrance spreads throughout the room. This happens because the perfume molecules move from an area of higher concentration to areas of lower concentration. This process is known as diffusion. Diffusion does not require any energy from the cell. It occurs naturally due to the continuous movement of particles. In living organisms, diffusion helps in the exchange of gases. Oxygen diffuses into cells, while carbon dioxide diffuses out of cells. Now let us discuss another important process called osmosis. Osmosis is a special type of diffusion. It refers specifically to the movement of water molecules through a selectively permeable membrane from a region of higher water concentration to a region of lower water concentration. In simple words, water moves from a dilute solution to a concentrated solution through a selectively permeable membrane. Remember this definition carefully because it is frequently asked in examinations. To understand osmosis better, let us look at the famous potato experiment mentioned in NCERT. In this experiment, a potato is hollowed out to form a cup-like structure. The cavity is filled with a concentrated sugar solution, and the potato is placed in a beaker containing plain water. After some time, the level of sugar solution inside the potato cavity rises. Why does this happen? The cells of the potato act as a selectively permeable membrane. Water from the beaker moves through the potato cells into the sugar solution because the sugar solution has a lower water concentration than the surrounding water. As a result, the volume of liquid inside the cavity increases. This demonstrates osmosis. Another example is the carrot experiment. A fresh carrot is placed in a concentrated salt solution. After some time, the carrot becomes soft and shrinks. This happens because water moves out of the carrot cells into the surrounding concentrated solution through osmosis. Now let us understand three important terms related to osmosis. The first is a hypotonic solution. A hypotonic solution contains a lower concentration of dissolved substances and a higher concentration of water compared to the cell. When a cell is placed in a hypotonic solution, water enters the cell. As a result, the cell swells. The second is a hypertonic solution. A hypertonic solution contains a higher concentration of dissolved substances and a lower concentration of water compared to the cell. When a cell is placed in a hypertonic solution, water moves out of the cell. As a result, the cell shrinks. The third is an isotonic solution. An isotonic solution has the same concentration of dissolved substances as the cell. In this condition, water moves into and out of the cell at equal rates. Therefore, there is no net change in the size of the cell. Students, these three terms are very important for understanding water balance in living organisms. Now let us quickly summarize the key points from this slide. The cell membrane is a thin, living, flexible boundary present around every cell. It is selectively permeable, allowing some substances to pass while restricting others. Diffusion is the movement of particles from higher concentration to lower concentration. Osmosis is the movement of water through a selectively permeable membrane from a region of higher water concentration to a region of lower water concentration. The potato and carrot experiments demonstrate osmosis in living tissues. Hypotonic solutions cause cells to gain water and swell. Hypertonic solutions cause cells to lose water and shrink. Isotonic solutions maintain a balance with no net movement of water. These concepts are fundamental for understanding how cells exchange materials with their surroundings and maintain life processes. Now let us study another important structure found in plant cells, the cell wall, and compare plant cells and animal cells. Let us begin with the cell wall. The cell wall is a rigid, non-living outer covering present outside the cell membrane in plant cells. It is mainly composed of a substance called cellulose. The cell wall provides strength, support, and protection to the cell. It helps plant cells maintain a definite shape and prevents them from bursting when excess water enters through osmosis. Unlike the cell membrane, the cell wall is freely permeable. This means that most substances can pass through it easily. One important point to remember is that animal cells do not possess a cell wall. They are surrounded only by a cell membrane. Now let us study the structure of a plant cell. A plant cell is usually rectangular or box-shaped because of the presence of a rigid cell wall. Inside the cell wall lies the cell membrane, which controls the movement of substances. The cytoplasm fills the interior of the cell and contains various organelles that perform specific functions. The nucleus acts as the control center of the cell and contains genetic material. Plant cells also contain chloroplasts, which are green-colored organelles responsible for photosynthesis. Photosynthesis is the process by which plants prepare their own food using sunlight, carbon dioxide, and water. Another characteristic feature of plant cells is the presence of a large central vacuole. This vacuole stores water, food, minerals, and waste materials. It also helps maintain the turgidity of the cell, keeping the plant upright and firm. Now let us look at the animal cell. Animal cells are generally round, oval, or irregular in shape because they lack a rigid cell wall. Like plant cells, animal cells also contain a cell membrane, cytoplasm, nucleus, mitochondria, ribosomes, endoplasmic reticulum, and Golgi apparatus. However, there are some important differences. Animal cells do not contain chloroplasts because animals cannot prepare their own food through photosynthesis. Animal cells also possess smaller vacuoles compared to the large central vacuole found in plant cells. In many animal cells, structures called centrioles are present, which play an important role during cell division. Now let us compare plant cells and animal cells more systematically. The first difference is the presence of a cell wall. Plant cells have a cell wall, whereas animal cells do not. The second difference is shape. Plant cells usually have a fixed rectangular shape, while animal cells often have an irregular shape. The third difference is chloroplasts. Chloroplasts are present in plant cells but absent in animal cells. The fourth difference is vacuoles. Plant cells generally contain one large central vacuole, while animal cells contain many small vacuoles or sometimes none at all. The fifth difference is food storage. Plants usually store food in the form of starch, whereas animals store food in the form of glycogen. Despite these differences, plant cells and animal cells share many common structures. Both possess a cell membrane, cytoplasm, nucleus, mitochondria, ribosomes, endoplasmic reticulum, and Golgi apparatus. These common organelles perform essential life functions in both types of cells. Now let us discuss two important laboratory activities mentioned in NCERT. The first activity involves observing onion peel cells. A thin peel is removed from the inner surface of an onion scale leaf and placed on a microscope slide. After adding a drop of water and stain, the cells can be observed under a microscope. Students can clearly see the rectangular shape of the cells, the cell wall, cell membrane, nucleus, and large vacuole. The second activity involves observing cheek cells. A cotton swab is gently rubbed on the inner surface of the cheek to collect cells. These cells are placed on a slide, stained, and observed under a microscope. Cheek cells appear irregular in shape and lack a cell wall. However, the cell membrane, cytoplasm, and nucleus can be clearly observed. These activities help us understand the structural differences between plant and animal cells. Students, remember these important exam points. The cell wall is present only in plant cells and is made mainly of cellulose. Plant cells contain chloroplasts and a large central vacuole. Animal cells do not contain a cell wall or chloroplasts. Plant cells are generally rectangular, whereas animal cells are usually irregular in shape. Both plant and animal cells possess a cell membrane, cytoplasm, nucleus, mitochondria, ribosomes, endoplasmic reticulum, and Golgi apparatus. The observation of onion peel cells and cheek cells provides practical evidence of the existence and structure of cells. With this understanding of plant cells and animal cells, we are now ready to study different types of cells and learn how prokaryotic and eukaryotic cells are organized in living organisms. Now we will study another important classification of cells based on the presence or absence of a true nucleus. All cells are broadly divided into two major categories, prokaryotic cells and eukaryotic cells. Understanding this classification is very important because it helps us understand the evolution and complexity of life on Earth. Let us begin with prokaryotic cells. The word prokaryotic comes from two Greek words. Pro means before and karyo means nucleus. Therefore, prokaryotic cells are cells that do not possess a true nucleus. These are the simplest and most primitive types of cells. In prokaryotic cells, the genetic material or DNA is not enclosed within a nuclear membrane. Instead, it is present freely in the cytoplasm in a region called the nucleoid. Another important feature of prokaryotic cells is that they lack membrane-bound organelles. This means structures such as mitochondria, Golgi apparatus, endoplasmic reticulum, and lysosomes are absent. Even though they lack these organelles, prokaryotic cells can perform all necessary life functions. Most prokaryotic organisms are unicellular, meaning they consist of only one cell. Examples include bacteria, cyanobacteria or blue-green algae, and mycoplasma. Because of their simple structure, prokaryotic cells are usually very small, ranging from about 0.1 to 5 micrometers in size. Their method of reproduction is also simple. They divide through a process called binary fission, where one cell divides into two identical daughter cells. Now let us move to eukaryotic cells. The word eukaryotic comes from the Greek words eu meaning true and karyo meaning nucleus. Therefore, eukaryotic cells possess a true nucleus surrounded by a nuclear membrane. This nucleus protects and organizes the genetic material of the cell. Unlike prokaryotic cells, eukaryotic cells contain several membrane-bound organelles such as mitochondria, endoplasmic reticulum, Golgi Apparatus, Lysosomes, Chloroplasts, and Vacuoles. These organelles perform specialized functions, making eukaryotic cells more efficient and complex. Eukaryotic cells are generally larger than prokaryotic cells and range from about 10 to 100 micrometers in size. They may be unicellular or multicellular. Examples of unicellular eukaryotes include amoeba, paramecium, and yeast. Examples of multicellular eukaryotes include plants, animals, and human beings. Now let us compare prokaryotic and eukaryotic cells. The first and most important difference is the nucleus. Prokaryotic cells lack a true nucleus, whereas eukaryotic cells possess a well-defined nucleus enclosed by a nuclear membrane. The second difference is the presence of membrane-bound organelles. Prokaryotic cells do not contain membrane-bound organelles, while eukaryotic cells contain many specialized organelles. The third difference is size. Prokaryotic cells are generally smaller, whereas eukaryotic cells are larger and more complex. The fourth difference is DNA organization. In prokaryotic cells, DNA is circular and naked, meaning it is not associated with proteins. In eukaryotic cells, DNA is linear and organized into chromosomes with the help of proteins. The fifth difference is cell division. Prokaryotic cells divide by binary fission, while eukaryotic cells divide by mitosis or meiosis. Students, remember that prokaryotic cells represent the earliest forms of life on Earth. Scientists believe that more complex eukaryotic cells evolved from simpler prokaryotic ancestors over millions of years. Now let us briefly discuss the cytoplasm. The cytoplasm is a jelly-like substance present between the cell membrane and the nucleus. It contains various organelles and serves as the site for numerous biochemical reactions. The cytoplasm acts as a medium through which nutrients, enzymes, and other substances move within the cell. Without cytoplasm, cellular activities cannot occur properly. Students, here are some important exam points to remember. Prokaryotic cells do not possess a true nucleus. Eukaryotic cells possess a well-defined nucleus. Prokaryotic cells lack membrane-bound organelles. Eukaryotic cells contain membrane-bound organelles. Examples of prokaryotes include bacteria and cyanobacteria. Examples of eukaryotes include amoeba, plants, animals, and humans. Prokaryotic cells divide by binary fission, whereas eukaryotic cells divide by mitosis and meiosis. Remember this simple rule. Simple cells without a true nucleus are called prokaryotic cells. Complex cells with a true nucleus are called eukaryotic cells. This classification forms the foundation for understanding all living organisms. In this slide, we will study the most important organelle of the cell, the nucleus, and understand how DNA, chromosomes, genes, and ribosomes work together to control life. Let us begin with the nucleus. The nucleus is often called the control center or brain of the cell because it regulates and coordinates all cellular activities. Just as the principal manages the activities of an entire school, the nucleus controls the activities of the entire cell. The nucleus is usually spherical or oval in shape and is surrounded by a double-layered membrane called the nuclear membrane or nuclear envelope. The nuclear membrane separates the contents of the nucleus from the cytoplasm and contains tiny openings called nuclear pores. These pores allow the exchange of materials between the nucleus and the cytoplasm. Inside the nucleus is a jelly-like substance called nucleoplasm. Suspended within the nucleoplasm are thread-like structures known as chromatin. The nucleus also contains a dense, round structure called the nucleolus. The nucleolus plays an important role in the formation of ribosomes, which are responsible for protein synthesis. One of the most important functions of the nucleus is storing and protecting the genetic material of the cell. This genetic material is known as DNA. DNA stands for deoxyribonucleic acid. DNA is often referred to as the hereditary material because it carries the information that is passed from parents to offspring. Every characteristic of an organism, such as eye color, hair type, height, skin color, and many other traits, is controlled by information stored in DNA. DNA has a unique double helix structure, which resembles a twisted ladder. This structure was explained by James Watson and Francis Crick in 1953. DNA is made up of smaller units called nucleotides. Each nucleotide consists of three components: a phosphate group, a deoxyribose sugar, and a nitrogenous base. There are four types of nitrogenous bases present in DNA. These are adenine, represented by the letter A, thymine, represented by T, guanine, represented by G, and cytosine, represented by C. These bases pair in a specific manner. Adenine always pairs with thymine, and guanine always pairs with cytosine. The sequence of these bases stores genetic information, much like letters form words and sentences in a book. Now let us understand chromatin and chromosomes. During most of the cell's life, DNA exists as long, thin, thread-like structures called chromatin. Chromatin is a combination of DNA and proteins. When a cell prepares to divide, the chromatin fibers become shorter, thicker, and highly condensed. At this stage, they appear as structures called chromosomes. Therefore, chromosomes are simply condensed forms of chromatin. Chromosomes are extremely important because they carry genetic information from one generation to the next. Humans possess 46 chromosomes arranged in 23 pairs. Students should remember this number because it is frequently asked in examinations. Now let us discuss genes. A gene is a specific segment of DNA that contains instructions for a particular characteristic or function. Genes are often called the units of heredity. For example, certain genes determine eye color, while others influence height, blood group, or hair texture. Thousands of genes are present on each chromosome. Together, these genes determine the characteristics of an organism. You can think of DNA as a large book, chromosomes as chapters in that book, and genes as individual instructions written within the chapters. Now let us move to ribosomes. Ribosomes are tiny cellular structures responsible for protein synthesis. They are often called the protein factories of the cell. Proteins are essential molecules required for growth, repair, enzyme production, hormone synthesis, and numerous other life processes. Ribosomes may be found freely floating in the cytoplasm or attached to the rough endoplasmic reticulum. Their main function is to read genetic instructions and use them to assemble proteins. Without ribosomes, cells would not be able to produce the proteins necessary for survival. This is why ribosomes are considered one of the most important structures within the cell. Students, let us quickly revise the key points from this slide. The nucleus is the control center of the cell. It contains DNA, which stores hereditary information. DNA is made up of nucleotides, and has a double helix structure. Chromatin condenses to form chromosomes during cell division. Chromosomes carry genes. Genes are the units of heredity and determine various characteristics of organisms. Ribosomes are the sites of protein synthesis and are often called the protein factories of the cell. Remember this simple sequence. DNA, genes, chromosomes, hereditary characters. This relationship is very important and frequently appears in examinations. Now we will explore the remaining cell organelles that work together like different departments of a highly organized factory. Each organelle has a specific function, and together they ensure the smooth functioning of the cell. Let us begin with the endoplasmic reticulum, commonly known as the ER. The endoplasmic reticulum is a network of membrane-bound tubes and channels spread throughout the cytoplasm. Its main function is to transport materials from one part of the cell to another. The ER acts like a highway system inside the cell, allowing substances to move efficiently. There are two types of endoplasmic reticulum. The first is rough endoplasmic reticulum or RER. RER has ribosomes attached to its surface, giving it a rough appearance under the microscope. Because ribosomes are present, the rough ER is involved in the synthesis and transport of proteins. The second type is smooth endoplasmic reticulum or SER. SER lacks ribosomes and therefore appears smooth. Its primary functions include the synthesis of lipids or fats, detoxification of harmful substances, and storage of calcium ions. In liver cells, the smooth ER plays an important role in breaking down drugs and toxic chemicals. Now let us move to the Golgi apparatus. The Golgi apparatus was discovered by the Italian scientist Camillo Golgi. It consists of a series of flattened membrane-bound sacs. The Golgi apparatus acts as the packaging and distribution center of the cell. Proteins and lipids produced by the endoplasmic reticulum are transported to the Golgi apparatus. The Golgi apparatus modifies, sorts, packages, and dispatches these materials to different parts of the cell or outside the cell. You can think of the Golgi apparatus as a post office that receives packages, labels them, and sends them to their correct destinations. Next, let us study lysosomes. Lysosomes are small membrane-bound sacs filled with powerful digestive enzymes. These enzymes help break down waste materials, worn-out organelles, foreign particles, and harmful substances. Lysosomes play an important role in cleaning and recycling cellular materials. Because they contain powerful digestive enzymes, lysosomes are sometimes called the suicide bags of the cell. If a cell becomes severely damaged, lysosomes may release their enzymes and digest the entire cell. This process helps protect the organism by removing damaged or unwanted cells. Now let us study one of the most important organelles, the mitochondria. Mitochondria are often called the powerhouses of the cell. Their main function is to produce energy through a process called cellular respiration. During cellular respiration, food molecules such as glucose are broken down in the presence of oxygen to release energy. This energy is stored in the form of ATP, which stands for adenosine triphosphate. ATP is often called the energy currency of the cell because it provides energy for all cellular activities. Without mitochondria, cells would not have sufficient energy to perform their functions. An interesting fact about mitochondria is that they contain their own DNA and can replicate independently within the cell. Now let us move to vacuoles. Vacuoles are membrane-bound sacs used for storage. They store water, food, minerals, pigments, and waste products. In plant cells, A large central vacuole occupies a significant portion of the cell. This vacuole helps maintain turgidity, which keeps plants upright and firm. When plants lose water, the vacuoles shrink, causing the plant to wilt. Animal cells generally possess smaller vacuoles compared to plant cells. Next, let us discuss plastids. Plastids are organelles found only in plant cells. There are three main types of plastids. The first type is chloroplasts. Chloroplasts contain a green pigment called chlorophyll. Chlorophyll absorbs sunlight and enables plants to carry out photosynthesis. During photosynthesis, plants use sunlight, carbon dioxide, and water to prepare food. Because of this function, chloroplasts are often called the food factories of plant cells. The second type is chromoplasts. Chromoplasts contain pigments that give flowers, fruits, and some plant parts their bright colors such as red, yellow, and orange. These colors help attract insects and animals for pollination and seed dispersal. The third type is leucoplasts. Leucoplasts are colorless plastids responsible for storing food materials such as starch, oils, and proteins. Students, let us now understand how all these organelles work together. The nucleus acts as the control center and gives instructions. Ribosomes manufacture proteins. The endoplasmic reticulum transports proteins and lipids. The Golgi apparatus modifies and packages these materials. Lysosomes clean and recycle cellular waste. Mitochondria provide energy in the form of ATP. Vacuoles store materials and maintain water balance. Chloroplasts prepare food through photosynthesis. Together, these organelles ensure that the cell remains alive, healthy, and functional. Let us quickly revise the important exam points. The endoplasmic reticulum is the transport system of the cell. The rough air synthesizes proteins, while the smooth air synthesizes lipids and detoxifies harmful substances. The Golgi apparatus packages and distributes cellular materials. Lysosomes contain digestive enzymes and are called suicide bags. Mitochondria are the powerhouses of the cell and produce ATP. Vacuoles store water, food, minerals, and waste products. Chloroplasts contain chlorophyll and perform photosynthesis. Chromoplasts provide color, while leucoplasts store food. Students, remember this simple analogy. The cell is like a factory. The nucleus is the manager. Ribosomes are the workers. The air is the transport network. The Golgi apparatus is the packaging department. Lysosomes are the cleaning staff. Mitochondria are the power station. Vacuoles are the storage rooms. And chloroplasts are the food production units. In the previous slide, we studied the different organelles present inside a cell and learned how they work together to keep the cell alive and functioning. Now we will study one of the most important biological processes, cell division. Cell division is the process by which a parent cell divides to produce new daughter cells. Every living organism begins life as a single cell. A tiny fertilized egg gradually divides again and again to form millions and eventually trillions of cells that make up a complete organism. Without cell division, growth, repair, reproduction, and continuity of life would not be possible. Let us first understand why cell division is necessary. As organisms grow, their cells increase in number through cell division. When tissues are damaged due to injury, old or damaged cells are replaced by new cells produced through cell division. In many organisms, cell division also plays a role in reproduction. Thus, cell division is essential for growth, repair, maintenance, and reproduction. There are two major types of cell division found in living organisms. The first is mitosis, also known as equational division. The second is meiosis, also known as reduction division. Let us begin with mitosis. Mitosis occurs in somatic or body cells. The main purpose of mitosis is growth, repair, and replacement of damaged cells. During mitosis, one parent cell divides to form two daughter cells. An important feature of mitosis is that both daughter cells are genetically identical to the parent cell. The chromosome number also remains unchanged. For example, if the parent cell contains 46 chromosomes, each daughter cell will also contain 46 chromosomes. This is why mitosis is called equational division. Now let us study the stages of mitosis. The first stage is interphase. Although not technically part of mitosis, it is the preparation stage. During interphase, the cell grows, carries out normal activities, and duplicates its DNA so that each daughter cell will receive a complete set of genetic material. The second stage is prophase. In prophase, chromatin fibers condense and become visible as chromosomes. The nuclear membrane begins to disappear and spindle fibers start forming. The third stage is metaphase. In metaphase, chromosomes arrange themselves along the center or equatorial plane of the cell. Spindle fibers attach to the chromosomes. The fourth stage is anaphase. During anaphase, sister chromatids separate and move toward opposite poles of the cell. This ensures that each future daughter cell receives an identical set of chromosomes. The fifth stage is telophase. In telophase, chromosomes reach opposite poles and begin to uncoil. New nuclear membranes form around each set of chromosomes. Finally, the cytoplasm divides in a process called cytokinesis, resulting in two identical daughter cells. Students, remember this sequence: interphase, prophase, metaphase, anaphase, telophase, cytokinesis. This order is very important for examinations. Now let us study meiosis. Meiosis occurs in reproductive organs such as testes and ovaries. Its main function is the formation of gametes, which are sperm cells and egg cells. Unlike mitosis, meiosis involves two successive divisions and produces 4 daughter cells. Another important difference is that the chromosome number is reduced to half. For example, if the parent cell contains 46 chromosomes, each daughter cell formed through meiosis will contain only 23 chromosomes. This is why meiosis is called reduction division. Meiosis is extremely important because it ensures that the chromosome number remains constant from one generation to the next. If meiosis did not occur, the chromosome number would double in every generation. An important event during meiosis is crossing over. During crossing over, homologous chromosomes exchange genetic material. This results in genetic variation among offspring. Because of genetic variation, no two individuals are exactly alike except identical twins. Now let us compare mitosis and meiosis. Mitosis occurs in body cells, whereas meiosis occurs in reproductive cells. Mitosis produces 2 identical daughter cells, whereas meiosis produces 4 genetically different daughter cells. Mitosis maintains chromosome number, whereas meiosis reduces chromosome number by half. Mitosis is important for growth and repair, whereas meiosis is important for sexual reproduction and genetic variation. Now let us discuss an important NCERT activity involving the onion root tip. Scientists often study cell division using onion root tips because cells in the root tip divide rapidly. When a stained onion root tip is observed under a microscope, different stages of mitosis can be clearly seen. This experiment provides direct evidence of cell division in living tissues. Students should remember this activity because it is commonly asked in practical examinations. Let us now briefly discuss cell culture. Cell culture is a laboratory technique in which cells are grown under controlled conditions outside the body. Scientists use cell culture for medical research, vaccine development, genetic studies, and disease investigation. Cell culture has become an important tool in modern biology and medicine. Now let us understand what happens when cell division goes wrong. Sometimes errors occur during mitosis. When the normal control of cell division is lost, cells continue dividing uncontrollably. This uncontrolled growth leads to the formation of tumors and may result in cancer. Cancer is therefore closely related to abnormal cell division. Errors during meiosis can also occur. Such errors may lead to an abnormal number of chromosomes in offspring, resulting in certain genetic disorders. This highlights the importance of accurate chromosome separation during cell division. Students, let us quickly revise the important exam points. Cell division is the process by which a parent cell produces new daughter cells. Mitosis is equational division and produces two genetically identical daughter cells. Meiosis is reduction division and produces four genetically different daughter cells. Mitosis is responsible for growth and repair. Meiosis is responsible for gamete formation and genetic variation. The onion root tip is commonly used to observe mitosis. Uncontrolled mitosis can lead to cancer. Remember this simple formula. Mitosis is equal to growth plus repair plus same chromosome number. Meiosis is equal to reproduction plus variation plus half chromosome number. We have reached the final slide of this chapter. In the previous slides, we explored the fascinating world of cells, learned about their structure, functions, organelles, and the process of cell division. Now it is time to revise the entire chapter in a systematic manner and understand the most important concepts that you should remember for examinations. Let us begin with cell theory. Cell theory is one of the most important concepts in biology because it explains the fundamental nature of living organisms. The first scientist associated with cell theory was Matthias Schleiden. In 1838, Schleiden proposed that all plants are made up of cells. A year later, in 1839, Theodor Schwanner extended this idea and stated that all animals are also made up of cells. Together, these observations formed the basis of cell theory. Later, in 1855, Rudolf Virchow added an important statement. He proposed that all new cells arise from pre-existing cells. This statement is often remembered as omnis cellula e cellula, which means every cell comes from another cell. According to cell theory, all living organisms are made up of cells. The cell is the basic structural and functional unit of life. All new cells arise from pre-existing cells. Students, these three statements are extremely important and frequently appear in examinations. Now let us quickly revise the entire chapter through a simple mind map. At the center of our chapter is the cell, the fundamental unit of life. Every living organism is made up of cells. Cells may be unicellular or multicellular. Unicellular organisms consist of a single cell, while multicellular organisms consist of many cells working together. Cells are broadly classified into two categories. The first category is prokaryotic cells. These cells lack a true nucleus and membrane-bound organelles. Examples include bacteria and cyanobacteria. The second category is eukaryotic cells. These possess a true nucleus and membrane-bound organelles. Examples include plants, animals, fungi, and protists. Every cell contains several important structures. The cell membrane acts as a selectively permeable boundary. The cytoplasm serves as the site for numerous biochemical reactions. The nucleus acts as the control center and contains DNA. DNA contains genes, which determine hereditary characteristics. DNA condenses to form chromosomes during cell division. We also studied various cell organelles. Mitochondria are the powerhouses of the cell because they produce ATP. Ribosomes synthesize proteins. The endoplasmic reticulum transports materials within the cell. The Golgi Apparatus packages and distributes cellular products. Lysosomes digest waste materials and are known as the suicide bags of the cell. Vacuoles store food, water, and waste materials. Chloroplasts carry out photosynthesis in plant cells. Students, another important concept discussed in this chapter was cell division. Cell division allows organisms to grow, repair damaged tissues, and reproduce. There are two major types of cell division. Mitosis produces two genetically identical daughter cells and helps in growth and repair. Meiosis produces four genetically different daughter cells and helps in reproduction and genetic variation. Remember this simple formula. Mitosis equals growth and repair. Meiosis equals reproduction and variation. Now let us discuss two additional biological concepts. The first is contact inhibition. Normally, cells stop dividing when they come into contact with neighboring cells. This mechanism prevents uncontrolled growth and helps maintain proper tissue organization. When contact inhibition fails, cells may continue dividing uncontrollably, which can contribute to cancer. The second concept is programmed cell death, also known as apoptosis. Not all cells are meant to live forever. Certain cells are programmed to die at specific stages of development or when they become damaged. This controlled cell death helps maintain the health and proper functioning of the organism. For example, during the development of human fingers, cells between the fingers undergo programmed cell death, allowing separate fingers to form. Now let us understand totipotency. Totipotency is the ability of a single cell to develop into an entire organism. Plant cells are especially known for their remarkable totipotency. Scientists use this property in plant tissue culture to produce new plants from a small piece of tissue. This technique has great importance in agriculture and biotechnology. Let us also learn about an important Indian scientist. Dr. Arun Kumar Sharma is regarded as one of India's pioneering cytogeneticists. His contributions greatly advanced the study of chromosomes and cell biology in India. Students preparing for higher studies in biology should remember his name. Now let us quickly revise the most important one-mark questions from this chapter. Who discovered cells? Robert Hooke in 1665. What is the basic structural and functional unit of life? The cell. What is the powerhouse of the cell? Mitochondria. Which organelle synthesize proteins? Ribosomes. Which organelle contains chlorophyll? Chloroplasts. What is the hereditary material of the cell? DNA. What is the control center of the cell? The nucleus. What is osmosis? The movement of water through a selectively permeable membrane from higher water concentration to lower water concentration. What is mitosis? Cell division producing two identical daughter cells. What is meiosis? Cell division producing four genetically different daughter cells with half the chromosome number. Students, Here are some final examination tips. Learn all diagrams carefully. Practice labeling plant cells, animal cells, mitochondria, chloroplasts, and the nucleus. Memorize important definitions exactly as given in NCERT. Understand the differences between plant and animal cells. Understand the differences between prokaryotic and eukaryotic cells. Understand the differences between mitosis and meiosis. Revise all NCERT activities, especially onion peel cells, cheek cells, osmosis experiments, and onion root tip observations. These topics are frequently tested in examinations. Before we conclude, let us summarize the chapter in one sentence. Cells are the fundamental units of life, containing specialized structures that work together to perform all activities necessary for the survival, growth, reproduction, and continuity of living organisms. Thank you, students, for joining this complete journey through the world of cells. Keep revising regularly, practice diagrams, solve NCERT questions, and strengthen your concepts. Remember, small cells, big life. Understand cells, understand life. See you in the next chapter. Happy learning with Rush Learning Hub.